So tonight's lecture deals with Chapter 22, the lymphatic system and immunity, and this begins on page 846. The lymphoid system contributes to homeostasis by draining interstitial fluid, as well as providing the mechanisms for defense against disease. The purpose of this chapter is to understand the lymphatic system structure and function, compare and contrast the innate and adaptive immune systems, compare and contrast cell-mediated and antibody-mediated immunity, and discuss the effects of stress and aging on immunity. The lymphatic system is part of the circulatory system and a vital part of the immune system, comprising a network of lymphatic vessels that carry a clear fluid called lymph directionally toward the heart. Unlike the cardiovascular system, the lymphatic system is not a closed system. The human circulatory system processes an average of 20 liters of blood per day through capillary filtration, which removes plasma while leaving the blood cells. Roughly 17 liters of the filtered plasma are reabsorbed directly into the blood vessel, while the remaining 3 liters remain in the interstitial fluid. One of the main functions of the lymph system is to provide an accessory return route to the blood for the surplus 3 liters. The other main function is that of defense in the immune system. Lymph is very similar to blood plasma. It contains lymphocytes and other white blood cells. It also contains waste product and debris of cells together with bacteria and protein. The function of the lymphatic system. The lymphatic system has multiple interrelated functions. First is the removal of interstitial fluid from tissues. Lymphatic vessels drain excess interstitial fluid from the tissue spaces and return it to the blood. This function closely links it with the cardiovascular system. In fact, without this function, the maintenance of circulating blood volume would not be possible. Secondly, it helps with the absorption and transportation of fatty acids and fats such as chyle from the digestive system. The lymphatic vessels transport lipids and lipid-soluble vitamins such as vitamin A, vitamin D, vitamin E, and vitamin K. Thirdly, it helps to transport white blood cells to and from the lymph nodes into the bones. So in this respect, it carries out immune responses. Lymphoid tissue initiates highly specific responses directed against particular microbes of abnormal cells. The lymph transport antigen-presenting cells to the lymph nodes where an immune response can be stimulated. Concerning disease resistance, nonspecific resistance or innate immunity is present at birth and includes defense mechanisms that provide general protection against invasion by a wide range of pathogens. And immunity, or more specifically adaptive immunity, this involves the activation of specific lymphocytes that combat a particular pathogen or a foreign substance. The body system that carries out immune responses is called the lymphatic system. The lymphatic system consists of several structures and organs that contain lymphatic tissue, bone marrow, and a fluid called lymph that flows within the lymphatic vessels. The associated organs composed of lymphoid tissue are the sites of lymphocyte production. Lymphocytes are concentrated in the lymph nodes. The spleen and thymus are also lymphoid organs of the immune system. The tonsils are lymphoid organs that are also associated with the digestive system. Furthermore, lymphoid tissues contain lymphocytes and also contain other types of cells for support. The system also includes all structures dedicated to the circulation and production of lymphocytes, which also includes the bone marrow and the lymphoid tissue associated with the digestive system. So this is figure 22.1 on page 848. The lymphoid system consists of lymph plasma, lymphatic vessels, lymphoid tissues, and red bone marrow. And we can see all of those features highlighted in green on this model. So as I've already talked about, the functions of the lymphatic system are as follows. It drains interstitial fluid, it transports dietary fats, and it carries out immune responses. Moving on to the next slide, the blood does not come into direct contact with the parenchymal cells and tissues in the body, but its constituents of blood first exit the microvascular exchange blood vessels to become interstitial fluid. Lymph is the fluid that is formed when interstitial fluid enters the initial lymphatic vessels of the lymphatic system. The lymph is then moved along the lymphatic vessel network by either intrinsic contractions of the lymphatic passages or by extrinsic compression of the lymphatic vessels via external tissue forces. An example of this would be the contraction of skeletal muscles. Eventually, the lymph vessels empty into the lymphatic ducts, which drain into one of the two subclavian veins near their junction with the internal jugular veins. Lymphatic vessels and capillaries. Lymphatic vessels begin as capillaries which are opened on one end. Lymphatic capillaries are located between the cells of many tissues. Lymphatic capillaries also merge to form lymphatic vessels which have thin walls and many valves. And this is figure 22.2 on page 849. And we can see that lymphatic capillaries are found throughout the body except in avascular tissues, the central nervous system, portions of the spleen, and bone marrow. And here at letter A, we see the relationship of capillaries to tissue cells and blood capillaries. And at letter B, we see the details of a lymphatic capillary. So lymphatic capillaries have greater permeability than blood capillaries and thus can absorb large molecules such as proteins and lipids. Lymphatic capillaries are also slightly larger in diameter than blood capillaries and have a unique one-way structure that permits interstitial fluid to flow into them but not out. The ends of the endothelial cells that make up the wall of a lymphatic capillary overlap. When the pressure is greater in the interstitial fluid than in the lymph plasma, the cells separate slightly, like the opening of a one-way swinging door, and interstitial fluid enters the lymphatic capillary. When the pressure is greater inside of the lymphatic capillary, the cells adhere more closely, and the lymph plasma cannot escape back into the interstitial fluid. The pressure is relieved as lymph plasma moves further down the lymphatic capillary. Attached to the lymphatic capillaries are anchoring filaments which contain elastic fibers. They extend out of the lymphatic capillary, attaching lymphatic endothelial cells to the surrounding tissues. When excess interstitial fluid accumulates and causes tissue swelling, the anchoring filaments are pulled, making the openings between the cells even larger so that more fluid can flow into the lymphatic capillary. In the small intestine, specialized lymphatic capillaries called lacteals carry dietary lipids into lymphatic vessels and ultimately into the blood. The presence of these lipids causes the lymph plasma draining from the small intestine to appear creamy white. Such lymph plasma is referred to as chyle. Elsewhere, lymph is a clear, pale yellow. Moving on to the next slide, lymph trunks and ducts. From the lymphatic vessels, lymph passes through the lymph nodes and into the lymph trunks. Lymph trunks include the lumbar, interstitial, bronchomediastinal, subclavian, and jugular trunks. Lymph trunks then merge to form either the thoracic duct or the right lymphatic duct. So this is figure 22.3 on page 850, and this shows routes for the drainage of lymph plasma from lymphatic trunks into thoracic right lymphatic ducts. So all lymph plasma returns to the bloodstream through the thoracic lymphatic duct and right lymphatic duct. So this is the same figure on page 851 now. And we can see a detailed anterior view of the thoracic and right lymphatic ducts. And this is figure 22.4 on page 852. And this is the formation and flow of lymph. So this schematic diagram shows the relationship of the lymphoid system to the cardiovascular system. And the arrows indicate the direction of flow of lymph plasma and blood. So the sequence of fluid flow is blood capillaries, or blood, then into the interstitial spaces, and this is for the interstitial fluid, the lymphatic capillaries for the lymph plasma, the lymphatic vessels, which also is for the lymph plasma, the lymphatic trunks or ducts, similarly for the lymph plasma, and then to the junction of the internal jugular and subclavian veins, where we're back to blood. Structure. The lymphatic system consists of lymphatic organs, a conducting network of lymphatic vessels, and the circulating lymph. The thymus and the bone marrow constitute the primary lymphoid organs involved in the production and early clonal selection of lymphocytic tissue. From the bone marrow, B cells immediately join the circulatory system and travel to secondary lymphoid organs in search of pathogens. T cells, on the other hand, travel from the bone marrow to the thymus, where they develop further. The central or primary lymphoid organs generate lymphocytes from immature progenitor cells. Moving on to the next slide. Secondary or peripheral lymphoid organs, which include lymph nodes and the spleen, maintain mature, naive lymphocytes and initiate an adaptive immune response. The peripheral lymphoid organs are the sites of lymphatic activation by antigens. Mature lymphocytes recirculate between the blood and the peripheral lymphoid organs until they encounter their specific antigen. The tertiary lymphatic tissue typically contains far fewer lymphocytes and assumes an immune role only when challenged with antigens that result in inflammation. It achieves this by importing the lymphocytes from the blood and the lymph. Moving on to the next slide, the lymphatic organs and tissues. The primary lymphatic organs are organs where immune cells become immunocompetent, and this includes the red bone marrow and the thymus. So this is figure 22.5 on 853. And we can see the thymus. They're situated medial to the lungs. The bilobed thymus is largest at puberty, and then the functional portion atrophies with age. And here we have the same figure and page number. And now we're just looking at a couple of light micrographs of the thymus. Moving on to the next slide. The thymus is a primary lymphoid organ and is the site of the maturation for the T cells, the lymphocytes of the adaptive immune system. The loss or lack of the thymus results in severe immunodeficiency and subsequent high susceptibility to infection. Let's talk now about secondary lymphatic organs and tissues. This includes the lymph nodes, the spleen, and the lymphatic nodules. The spleen synthesizes antibodies in its white pulp and removes antibody-coated bacteria and antibody-coated blood cells by the way of blood and lymph node circulation. Like the thymus, the spleen has only efferent lymphatic vessels. Both the short gastric arteries and the splenic artery supply it with blood. So this is figure 22.7 on page 857, and this shows the structure of the spleen. The spleen is the largest single mass of lymphoid tissue in the body. Moving on, lymph nodes. A lymph node is an organized collection of lymphoid tissue through which lymph passes on its way back to the blood. Lymph nodes are located at intervals along the lymphatic system. Several afferent lymph vessels bring in lymph, which percolates through the substance of the lymph node and is then drained out by an efferent lymph vessel. So you should remember from several slides ago, afferent means toward, efferent means away from. There are between 500 and 600 lymph nodes in the human body, many of which are grouped in clusters in different regions, as in the underarm and the abdominal areas. Lymph node clusters are commonly found at the base of the limbs and in the neck, where the lymph is collected from regions of the body likely to sustain pathogen contamination from injuries. The structure of a lymph node. So this is figure 22.6 on page 855. And as I already said, lymph nodes are present throughout the body and are usually clustered in groups. And this is figure 22.6 continued, but now we're on page 856. So you can see the micrograph of a lymph node. And then there on the right at letter C, we see the anterior view of the inguinal lymph node. Lymphatic nodules. Lymphatic nodules are masses of lymphatic tissue that are not surrounded by a capsule. They are scattered throughout the lamina propria of the mucous membranes, lining the gastrointestinal tract, urinary tract, and reproductive tracts, and they also line the respiratory airways. Lymphatic nodules in these areas are also referred to as mucosa-associated lymphatic tissue. As I said on the previous slide, lymph nodes are particularly numerous in the mediastinum, in the chest, neck, pelvis, axilla, inguinal region, and in association with the blood vessels of the intestines. The development of lymphatic tissue. This is figure 22.8 on page 858. Lymphoid tissues are derived from the mesoderm. Lymphoid tissues begin to develop by the end of the 5th week of embryonic life. Lymphatic vessels develop from lymph sacs that arise from the developing veins, which are derived from the mesoderm. The first lymph sacs to appear are the paired jugular lymph sacs at the junction of the internal jugular and subclavian veins. From the jugular lymph sacs, the lymphatic capillary plexuses spread to the thorax, upper limbs, neck, and head. Some of the plexuses enlarge to form lymphatic vessels in their respective regions. Each jugular lymph sac retains at least one connection with its jugular vein, the left one developing into the superior portion of the thoracic duct. The next lymph sac to appear is the unpaired retroperitoneal lymph sac. At the root of the mesentery of the intestine, it develops the primitive vena cava and mesenteric veins. Capillary plexuses and lymphatic vessels spread from the retroperitoneal lymph sac to the abdominal viscera and diaphragm. The sac establishes connections with the cisterna chyli, but loses its connections with neighboring veins. At about the time the retroperitoneal lymph sac is developing, another lymph sac, the cisterna chyli, develops inferior to the diaphragm on the posterior abdominal wall. It gives rise to the inferior portion of the thoracic duct and the cisterna chyli of the thoracic duct. Like the retroperitoneal lymph sac, the cisterna chyli also loses its connections with the surrounding veins. The last of the lymph sacs, the paired posterior lymph sacs, develop from the iliac veins. The posterior lymph sacs produce capillary plexuses and lymphatic vessels of the abdominal wall, pelvic region, and lower limbs. The posterior lymph sacs join the cisterna chyli to lose their connections with the adjacent veins. With the exception of the anterior part of the sac from which the cisterna chyli develops, all lymph sacs become invaded by mesenchymal cells and are converted into groups of lymph nodes. The spleen develops from the mesenchymal cells between the layers of the dorsal mesentery of the stomach. The thymus arises as an outgrowth of the third pharyngeal pouch. Moving on to the next slide, innate immunity. Innate immunity refers to a wide variety of body responses that serve to protect us against the invasion of a wide variety of pathogens and their toxins. We are born with this kind of immunity, and there's two lines of defense, the skin and mucous membranes, and some internal defenses. So the innate immunity includes the external physical and chemical barriers provided by the skin and mucous membranes. It also includes various internal defenses such as antimicrobial substances, natural killer cells, phagocytes, inflammation, and fever. The skin and mucous membranes. So it provides some mechanical defense such as the skin itself, mucous membranes, tears, saliva, mucus, cilia, epiglottis, urine flow, defecating, and vomiting. And they also provide some chemical defenses such as the sebum, lysozymes, and gastric juices. Continuing on with the innate immune system, we'll talk now about internal defenses, and this is figure 22.9 on page 861. And some internal defenses include antimicrobial proteins, phagocytes, natural killer cells, inflammation, and fever. Moving on to the next slide, this is also figure 22.9 on page 861. But now we have a cartoon of a phagocyte, and the major types of phagocytes are neutrophils and macrophages. And in this diagram, they go through the phases of phagocytosis. So you see there at step 1, we've got this chemotaxis, and this is where phagocytosis begins with a chemically stimulated movement of the phagocyte to the site of damage. Chemicals that attract phagocytes might come from invading microbes, white blood cells, damaged tissue cells, or activated complement proteins. Secondly, there's adherence. This is the attachment of the phagocyte to the microbe or other foreign material. Thirdly, there's ingestion. So the plasma membrane of the phagocyte extends projections called pseudopods that engulf the microbe in a process called ingestion. When the pseudopods meet, they fuse, surrounding the microorganism with a sac called a phagosome. At step 4, we go into the digestion phase. The phagosome enters the cytoplasm and merges lysosomes to form a single layer structure called a phagolysosome. The lysosome contributes lysozyme, which breaks down microbial cell walls and other digestive enzymes that degrade carbohydrates, proteins, lipids, and nucleic acid. The phagocyte also forms lethal oxidants such as superoxide anion, hypochlorite anion, and hydrogen peroxide in a process called oxidative burst. And finally, step 5, killing. The chemical onslaught provided by the lysozyme, digestive enzymes, and oxidants within a phagolysozyme quickly kills many cells of the microbes. Any materials that cannot be degraded further remain in structures called the residual bodies. And this is figure 22.10 on page 862. This details inflammation. So there's 3 stages of inflammation. That's vasodilation and increased permeability of blood vessels. Phagocytic emigration, remember we talked about that in a previous chapter, and tissue repair. So this brings us to table 22.1 on page 863, and this just summarizes the innate defenses. So I'll let you go through this table on your own time. And this next slide is also table 22.1 continued, so please review this on your own time. Moving on to adaptive immunity. Adaptive immunity is the ability of the body to defend itself against specific invading agents. Antigens are substances recognized as foreign that provoke immune responses. Adaptive immunity has both specificity and memory and is divided into 2 types: cell-mediated and antibody-mediated. And here we see figure 22.11 on page 865. And this shows how the B cells and pre-T cells arise from the pluripotent stem cells in the red bone marrow. B cells and T cells develop in primary lymphoid tissues and are activated in secondary lymphoid organs and tissues, such as the lymph nodes, spleen, and lymphatic nodules. Once activated, each type of lymphocyte forms a clone of cells that can recognize specific antigens. For simplicity, antigen receptors, such as CD4 proteins and CD8 proteins, are not shown in the plasma membranes of the cells of the lymphatic clones. And then, as you know from the previous slide, there are 2 types of adaptive immunity, and those are cell-mediated immunity, which you see there on the left side, and the antibody-mediated immunity, which you see on the right side. Antigens and antigen receptors. Antigens have 2 important characteristics: immunogenicity and reactivity. Entire microbes, or just part of microbes, may act as antigens. Typically, just certain small parts of a large antigen molecule act as the triggers for immune responses. These parts are called epitopes. So immunogenicity is the ability to provoke an immune response by stimulating the production of specific antibodies, the proliferation of specific T cells, or both. The term antigen derives from its function as an antibody generator. On the other hand, reactivity is the ability of the antigen to react specifically with the antibodies or cells it provoked. Strictly speaking, immunologists define antigens as substances that have reactivity. Substances with both immunogenicity and reactivity are considered complete antigens. Commonly, however, the term antigen implies both immunogenicity and reactivity, and we use the word in this way. Moving on to the next slide, this is figure 22.12 on page 866. So most antigens have several epitopes that induce the production of different antibodies or activate different T cells. So typically, just certain small parts of a large antigen molecule act as the trigger for the immune response. These small parts are called epitopes, or antigenic determinants. Most antigens have many epitopes, each of which induces production of a specific antibody or activates a specific T cell. Pathways of antigen processing. For an immune response to occur, B cells and T cells must recognize that a foreign antigen is present. B cells can recognize and bind to antigens in lymph, interstitial fluid, or blood plasma. On the other hand, T cells only recognize fragments of antigenic proteins that are processed and presented in a certain way. Moving on to the next slide, in antigen processing, antigenic proteins are broken down into peptide fragments that associate with the MHC molecules. And MHC stands for Major Histocompatibility Complex. The antigen MHC complex is then inserted into the plasma membrane of a body cell. This process is called antigen presentation. And we see this figure 22.13 on page 868. This is the processing and presenting of exogenous antigen by antigen-presenting cells. And we see that fragments of exogenous antigens are processed and then presented with the MHC2 complex molecules on the surface of an antigen-presenting cell. So at step 1, we have the ingestion of the antigen. The antigen-presenting cells ingest exogenous antigens by phagocytosis or endocytosis. Ingestion could occur almost anywhere in the body where invaders such as microbes have penetrated the innate defenses. Secondly, the digestion of the antigen into peptide fragments. Within the phagosome or endosome, protein-digesting enzymes split large antigens into short peptide fragments. Thirdly, synthesis of the MHC2 complex molecules. At the same time, the antigen-presenting cell synthesizes MHC2 molecules at the endoplasmic reticulum. Fourthly, packaging of the MHC2 molecules. Once synthesized, the MHC2 molecules are packaged into vesicles. Next, at number 5, fusion of vesicles. The vesicles containing antigen peptide fragments and MHC2 complex molecules merge and fuse. At the 6th step, we bind the peptide fragments to the MHC2 molecules. After fusion of the two types of vesicles, antigen peptide fragments bind to the MHC2 molecules. And at the 7th step, insertion of antigen MHC2 complexes into the plasma membrane. The combined vesicle that contains antigen MHC2 complexes undergoes exocytosis. As a result, the antigen MHC2 complexes are inserted into the plasma membrane. After processing an antigen, the antigen-presenting cell migrates to the lymphatic tissue to present the antigen to the T cells. Within the lymphatic tissue, a small number of T cells have compatibly shaped receptors and recognize and bind to antigen fragment MHC2 complexes. This triggers an adaptive immune response. The presentation of exogenous antigens together with the MHC2 molecules by antigen-presenting cells informs T cells that intruders are present in the body and that combative action should be taken. So moving on to page 869, figure 22.4, we can see the processing and presenting of endogenous antigens by an infected body cell. So at step 1, we've got digestion of the antigen into peptide fragments. Within the infected cell, protein-digesting enzymes split the endogenous antigen into short peptide fragments. Secondly, the synthesis of the MHC1 molecules. At the same time, the infected cell synthesizes the MHC1 molecules at the endoplasmic reticulum. Thirdly, the binding of peptide fragments to the MHC1 molecules, the antigen peptide fragments enter the endoplasmic reticulum and then bind to the MHC1 molecules. Fourthly, packaging of the antigen MHC1 molecules. From the ER, antigen MHC1 molecules are packaged into vesicles. And finally, at step 5, the insertion of antigen MHC1 complexes into the plasma membrane. The vesicles that contain the antigen MHC1 complexes undergo exocytosis. As a result, the antigen MHC1 complexes are inserted into the plasma membrane. So it should be noted that most cells of the body can process and present endogenous antigens. The display of endogenous antigen bound to an MHC1 molecule signals that a cell has been infected and needs help. Moving on to table 22.2 on page 869. So this is a summary of cytokines participating in immune responses. So please review this table on your own time. Let's move on to cell-mediated immunity. In cell-mediated immunity, an antigen is recognized and bound. A small number of T cells proliferate and differentiate into a clone of effector cells, and then the antigen is eliminated. A cell-mediated immune response begins with activation of a small number of T cells by a specific antigen. Once a T cell has been activated, it undergoes clonal selection. Recall that clonal selection is the process by which a lymphocyte proliferates and differentiates in response to a specific antigen. The result of clonal selection is the formation of a clone of cells that can recognize the same antigen as the original lymphocyte. Some of the cells of a T cell clone become effector cells, while other cells of the clone become memory cells. The effector cells of a T cell clone carry out immune responses that ultimately result in elimination of the intruder. And here we see figure 22.15, page 871, the activation and clonal selection of a helper T cell. So once a helper T cell is activated, it forms a clone of active helper T cells and memory helper T cells. Active helper T cells secrete a variety of cytokines. One very important cytokine produced by helper T cells is interleukin-2, which is needed for virtually all immune responses and is the prime trigger of T cell proliferation. On the other hand, the memory helper T cells of a helper T cell clone are not active cells. However, if the same antigen enters the body again in the future, memory helper T cells can quickly proliferate and differentiate into more active helper T cells and more memory helper T cells. And on this next figure, 22.16 on page 872, we see the activation and clonal selection of cytotoxic T cells. So once a cytotoxic T cell is activated, it forms a clone of active cytotoxic T cells and memory cytotoxic T cells. So active cytotoxic T cells attack other body cells that have been infected with the antigen. And then memory cytotoxic T cells do not attack infected body cells. Instead, they can quickly proliferate and differentiate into more active cytotoxic T cells and more memory cytotoxic T cells if the same antigen enters the body at a future time. The activity of cytotoxic T cells. After delivering a lethal hit, a cytotoxic T cell can detach and attack another infected target cell displaying the same antigen. So cytotoxic T cells release granzymes that trigger apoptosis and perforin that triggers cytolysis of infected target cells. Moving on to antibody-mediated immunity. In antibody-mediated immunity, an antigen is recognized and bound. Helper T cells co-stimulate the B cell, so the B cell can proliferate and differentiate into a clone of effector cells that produce antibodies. And then the antigen is eliminated. So here we see at figure 21.18 on page 874, the activation and clonal selection of B cells. So during activation of a B cell, an antigen binds to B cell receptors. These integral transmembrane proteins are chemically similar to antibodies that eventually are secreted by plasmacytes. Although B cells can respond to an unprocessed antigen present in lymph plasma or interstitial fluid, their response is much more intense when they process the antigen. Antigen processing in a B cell occurs in the following way. The antigen is taken into the B cell, broken down into peptide fragments, and combined with the MHC2 self-antigens, and moved to the B cell plasma membrane. Then helper T cells recognize the antigen MHC2 complex and deliver the co-stimulation needed for B cell proliferation and differentiation. The helper T cell produces interleukin-2 and other cytokines that function as co-stimulators to activate B cells. Once activated, a B cell undergoes clonal selection. As a result of this formation of a clone of B cells that consists of plasmacytes and memory B cells, plasma cells secrete antibodies. A few days after exposure to an antigen, plasmacytes secrete hundreds of millions of antibodies each day for about 4 or 5 days until the plasmacyte dies. Most antibodies travel in lymph plasma and blood plasma to the invasion site. Interleukin-4 and interleukin-6, also produced by helper T cells, enhance B cell proliferation, B cell differentiation into plasmacytes, and the secretion of antibodies by plasmacytes. Memory B cells do not secrete antibodies. Instead, they can quickly proliferate and differentiate into more plasmacytes and more memory B cells should the same antigen reappear at a future time. Different antigens stimulate different B cells to develop into plasmacytes and their accompanying memory B cells. All of the B cells of a particular clone are capable of secreting only one type of antibody, which is identical to the antigen receptor displayed by the B cell that first responded to it. Each specific antigen activates only those B cells that are predestined to secrete antibody specific to that antigen. Antibodies produced by a clone of plasmacytes enter the circulation and form an antigen-antibody complex with the antigen that initiated their production. So moving on to figure 22.19, page 875. We're talking about antibody structure, and antibodies belong to a group of glycoproteins called globulins. And for this reason, they are also known as immunoglobulins. Most antibodies contain 4 polypeptide chains. 2 of the chains are identical to each other and are called heavy chains. Each consists of about 450 amino acids. Short carbohydrate chains are attached to each heavy polypeptide chain. The 2 other polypeptide chains, also identical to each other, are called light chains, and each consists of about 220 amino acids. A disulfide bond holds each light chain to a heavy chain. 2 disulfide bonds also link the mid-region of the 2 heavy chains. This part of the antibody displays considerable flexibility and is called the hinge region. Because the antibody, quote-unquote, arms can move somewhat as the hinge region bends, an antibody can assume either a T shape or a Y shape. Beyond the hinge region, parts of the two heavy chains form the stem region. So we see on page 876, table 22.3, and this goes through the classes of immunoglobulins. And by this table, you can see that there are 5 of those: IgG, IgA, IgM, IgD, and IgE. So I'll let you go through this table on your own time. Moving on to the next slide, figure 22.20 on page 877. This shows the complement activation and results. And C stands for complement and the complement system. The complement system is a defensive system made up of over 30 proteins produced by the liver and found in circulating blood plasma and within tissues throughout the body. Collectively, the complement proteins destroy microbes by causing phagocytosis, cytolysis, and inflammation. They also prevent excessive damage to body tissues. So when activated, complement proteins enhance phagocytosis, cytolysis, and inflammation. So at step 1, an inactivated C3 splits into an activated C3a and C3b. At step 2, C3b binds to the surface of a microbe and receptors on phagocytes attached to the C3b. Thus, the C3b enhances phagocytosis by coating a microbe, a process called opsonization. Opsonization promotes the attachment of a phagocyte to a microbe. Thirdly, C3b also initiates a series of reactions that bring about cytolysis. First, C3b splits C5. The C5b fragment then binds to C6 and C7, which attach to the plasma membrane of an invading microbe. Then the C8 and several C9 molecules join other complement proteins together to form a cylinder-shaped membrane attack complex, which inserts into the plasma membrane. Fourthly, the membrane attack complex creates channels of the plasma membrane that result in cytolysis, the bursting of the microbial cells due to the inflow of extracellular fluid through the channels. And finally, at step 5, C3 and C5a bind to mast cells and cause them to release histamine that increases blood vessel permeability during inflammation. C5a also attracts phagocytes to the site of inflammation, and this is called chemotaxis. So here we see figure 22.21 on page 878. And the immunological memory is the basis for successful immunization and vaccination. So a hallmark of immune response is memory for specific antigens that have triggered immune responses in the past. Immunologic memory is due to the presence of long-lasting antibodies and very long-lived lymphocytes that arise during clonal selection of antigen-stimulated B cells and T cells. Immune responses, whether cell-mediated or antibody-mediated, are much quicker and more intense after a second or subsequent exposure to an antigen than after the first exposure. Initially, only a few cells have the correct specificity to respond, and the immune response may take several days to build the maximum intensity. Because thousands of memory cells exist after an initial encounter with an antigen, the next time the same antigen appears, they can proliferate and differentiate into helper T cells, cytotoxic T cells, plasmacytes within hours. And that's exactly what this graph shows. The secondary response to an antigen is more intense. And table 22.4 talks about ways to acquire adaptive immunity, and this is on page 879. So please spend a little bit of time on this slide by yourself. Self-recognition and self-tolerance. T cells undergo both positive and negative selection to ensure that they can recognize the self-MHC antigens and that they do not react to other self-proteins. So here we have figure 22.22 on page 879, and this talks about the development of self-recognition and the development of self-tolerance. So in order to function properly, your T cells must have two traits. They must be able to recognize your own major histocompatibility MHC complex proteins. This is a process known as self-recognition. And secondly, they must lack reactivity to peptide fragments from your own proteins. And this is a condition known as self-tolerance. And this part of the figure, these first two boxes or rectangles, they deal with the T cell. And the T cell has two types of selection. We have the positive selection, which is there at the top, and the negative selection, which is at the bottom. So in positive selection, some pre-T cells express T cell receptors that interact with the self-MHC proteins on epithelial cells in the thymic cortex. Because of this interaction, the T cells can recognize the MHC part of an antigen-MHC complex. These T cells will survive. Other immature T cells that fail to interact with the thymic epithelial cells are not able to recognize the self-MHC proteins, and these cells will undergo apoptosis. And then the development of self-tolerance occurs by a weeding out process, and this is called negative selection. And this is where the T cells interact with nodular dendritic cells located at the junction of the cortex and the medulla in the thymus. In this process, T cells with receptors that recognize self-peptide fragments or other self-antigens are eliminated or inactivated. The T cells selected to survive do not respond to self-antigens, the fragments of molecules that are normally present in the body. Negative selection occurs both via deletion and anergy. Anergy is the absence of a normal immune response to a particular antigen or allergen. In deletion, the self-reactive T cells undergo apoptosis and die. In anergy, they remain alive but are unresponsive to antigenic stimulation. B cells also develop tolerance through deletion and anergy. And this slide is also figure 22.22, but this is the bottom third of this figure. And this shows the development of self-recognition and self-tolerance concerning the B cell. So it should be pointed out that B cells also develop tolerance through deletion and anergy. While B cells are developing in the bone marrow, those cells exhibiting antigen receptors that recognize common self-antigens are deleted. Once B cells are released into the blood, however, anergy appears to be the main mechanism for preventing responses to self-proteins. When B cells encounter an antigen not associated with an antigen-presenting cell, the necessary co-stimulation signal often is missing. In this case, the B cell is likely to become anergic, or in other words, inactivated rather than activated. And now on this next slide, we see table 22.5 on page 881. And 22.5 summarizes the activities of the cells involved in adaptive immune responses. So please study this table on your own time. Moving on to the next slide, stress and immunity. Psychoneuroimmunology, or PNI, is a field that deals with common pathways that link the nervous system, endocrine, and immune systems. Research in this field indicates that thoughts, feelings, moods, and beliefs influence your level of health and the course of disease. Moving on to aging and the immune system. Aging results in the increased susceptibility to infections and malignancies, the increased production of autoantibodies, the decreased response to vaccines, and an overall decrease in immune system function. So moving on to disorders and homeostatic imbalances. Lymphadenopathy refers to one or more enlarged lymph nodes. Small groups or individually enlarged lymph nodes are generally reactive in response to infection or inflammation. This is called local lymphadenopathy. When many lymph nodes in different areas of the body are involved, this is called generalized lymphadenopathy. Moving on to the next slide. Lymphedema is a swelling caused by the accumulation of lymph. This may occur if the lymphatic system is damaged or has malformations. It usually affects the limbs, though the face, neck, and abdomen may also be affected. In an extreme state, called elephantitis, the edema progresses to the extent that the skin becomes thick, with an appearance similar to the skin on an elephant's limbs. Immunodeficiency. Immunodeficiency occurs when one or more of the components of the immune system are inactive. The ability of the immune system to respond to pathogens is diminished in both the young and the elderly, with immune responses beginning to decline at around 50 years. Obesity, alcoholism, and drug use are common causes of poor immune function. However, malnutrition is the most common cause of immunodeficiency in developing countries. Additionally, the loss of the thymus at an early age through genetic mutation or surgical removal results in severe immunodeficiency and a high susceptibility to infection. So immunodeficiencies can be inherited or acquired. Chronic granulomatous disease. This is where phagocytes have the reduced ability to destroy pathogens. This is an example of an inherited or congenital immunodeficiency. AIDS and some types of cancer can cause acquired immunodeficiency. Autoimmunity. Overactive immune responses comprise the other end of the immune dysfunction spectrum, particularly the autoimmune disorders. In the autoimmune disorders, the immune system fails to properly distinguish between the self and non-self, and then it attacks a part of your own body. So it seems that in the case of this particular disorder, autoimmunity, that the body loses the ability to self-recognize and to self-tolerate, as we were talking several slides ago. Hypersensitivity is an immune response that damages the body's own tissue. They are divided into 4 classes based on the mechanisms involved and the time course of the hypersensitivity reaction. So type 1 hypersensitivity is an immediate or anaphylactic reaction, often associated with an allergy. Symptoms can range from mild discomfort to death. Type 2 hypersensitivity occurs when antibodies bind to antigens on the patient's own cells, marking them for destruction. This is also called the antibody-dependent hypersensitivity. Type 3 hypersensitivity involves reactions. And type 4 hypersensitivity, also known as cell-mediated or delayed type hypersensitivity, usually takes between 2 and 3 days to develop. Type 4 reactions are involved in many autoimmune and infectious diseases, but may also involve contact dermatitis, for example, poison ivy. Moving on to the next slide, we'll start to talk about cancer now. And so the study of lymphatic drainage of various organs is important in the diagnosis, prognosis, and treatment of cancer. The lymphatic system, because of its physical proximity to many tissues of the body, is responsible for carrying cancer cells between the various parts of the body in a process called metastasis. The intervening lymph nodes can trap the cancer cells. If they are not successful in destroying the cancer cells, the nodes may also become sites of secondary tumors. Next slide. Cancer of the lymphatic system can be primary or secondary. Lymphoma refers to cancer that arises from lymphatic tissue. They are called leukemia when it is in the blood or marrow, and lymphoma when in the lymphatic tissue. They are grouped together under the name lymphoid malignancy. And on this next slide, we see our focus on homeostasis, and this is on page 883. So this talks about the contributions of the lymphoid system and immunity for all the body systems. B cells, T cells, and antibodies protect all body systems from attack from harmful foreign invaders, also called pathogens, foreign cells, and other cancer cells. Moving on to homeostatic imbalances, HIV/AIDS. Acquired immunodeficiency syndrome, AIDS, is a condition in which a person experiences an assortment of infections due to the progressive destruction of the immune system cells by the human immunodeficiency virus. And there on the right, you see figure 22.23 on page 882. And it's saying that the HIV virus has a diameter of about 100 to 140 nanometers. And this is what we call a retrovirus. And you can see that, like all viruses, it's just basically a protein coat and some sort of nucleic acid. And in this case, we see that the HIV virus is what we call an RNA virus. Homeostatic imbalances, allergies. Allergies occur when a person is overly reactive to a substance that is well tolerated by most others. When an allergic reaction occurs, so does tissue damage. There are 4 types of hypersensitive reactions, type 1 through 4, and we talked about that several slides ago. Homeostatic imbalances, autoimmune diseases. We also talked about this several slides ago, but let's go through it. An autoimmune disease occurs when the immune system fails to display self-tolerance and instead attacks the person's own body tissues. And that concludes tonight's lecture.